Hello everyone and welcome back to this online NPTEL certification course on aquatic geochemistry. In this lecture we will be discussing about the water sampling methods and different types of analysis that is done especially to understand water quality and a lot of different process based understandings. So, there are two types of data that is available for use in our hydrology, and that is firstly, of course, as you already must be knowing, is remotely sensed data. Okay. Now, what is remotely sensed data? You actually do not go to the field or do any kind of measurements you will have, but rather you will have satellites who will be as they are moving all across the globe, they will be taking the measurements. Okay. So, there is no direct physical contact, there is no direct measurement of that particular object. The satellites, they will have particular sensors and they will be taking the measurements. Okay. Now, what is the biggest advantage of remotely sensed data is that we can cover huge areas with the minimum physical effort. So, generally what happens in ground based studies and when there is a lot of field work involved, that we are always hindered due to logistical support. Logistical support means we have to provide manpower, there has to be all types of accommodation, traveling, collection of water samples, then bringing them back to the lab, doing the analysis and all that. All that can be extremely sometimes can become challenging due to multiple reasons. Okay. But and if we are targeting covering huge areas, then it becomes more difficult to convey the to conduct such kind of ground surveys. So, remotely sensed data is of extremely helpful in this particular use, that in this regard, that you can without visiting actually that particular location, you can get some data. And generally these satellite missions, they occur every they are occurring at a particular time interval. So, every month or every year, and there are many satellites that even can give you day data, every day data. So, daily data sets, and sometimes it can be even very short time resolution like hourly data and things like that. So, based on what you are, what parameter you are interested in, you will find some time scales. Depending on that, you have to find the best time scale, that is the best time resolution that you will be looking into, and the most useful data set you will be using for your own study. And so, first of all, remotely sensed data gives us access to huge areas. It helps us overcome the limitations due to lack of manpower, lack of financial support and all that. And it is also extremely useful when the locality of that particular place is very difficult to access. And so for example, if you want to conduct a yearly study somewhere in the Arctic, right. Now, of course, now we have all the stations in the Arctic regions and the Antarctic regions, and people are now staying there all throughout the year and all. But previously it was a extremely difficult job, and it is still difficult to access certain locations. And not only the colder regions, the polar regions, but even in the Himalayas, if you see, many locations are still inaccessible. That is, if you have to go there, you have to trek, you may be trekking for multiple days, and it becomes difficult to collect samples and maintain a regular visit. So, suppose you want to do that every month, maybe just forget about every day, maybe doing a monthly data collection will become very difficult if you always have to trek a few days. right. So, remote sensing data becomes very useful in these kind of cases. That means, it gives us accessibility to places where accessibility is very low, and sometimes it can also happen that that place is totally closed due to something, like maybe some extreme event has happened, maybe there has been a landslide, maybe there has been a flood event that has actually washed away the roads and all that, and therefore, to build more new roads and get accessibility to the place can take several months or even years. So, then what remote sensing data becomes very useful, and it is really helpful in those cases. Now, what is the disadvantage? The disadvantage is that it can actually quantify only those particular parameters that has a optical sensitivity, right. So, mostly remote sensing data will give us a lot of weather data, temperature sensitivity. So, sensors which can which has been developed to detect changes even from without being in physical contact, those kind of changes only can be detected. So, things like temperature of the atmosphere and things like that, and in terms of water only few parameters that are optically sensitive. So, for example, you will all often find discharge data will be there, okay. Discharge data will be there, sometimes you will see turbidity data is there, chlorophyll A data is there. So, these kind of data sometimes will be available with the remotely sensed data sets. Also, what will happen is that the spatial resolution sometimes can become a problem. Why? Because when so of course, the satellite is passing, okay, and the area that is covered, okay, that is called the spatial resolution. So, the spatial resolution can vary from few tens of kilometers to maybe 1 kilometer or 2 kilometer. That is the level of spatial resolution that you can receive. But if you want to go through to a much shorter resolution, you will not be able to do that. You will always be limited by what is the spatial resolution of that particular satellite mission is. So, that is one limitation if you think about, and of course, it can have it is limited where there is subsurface coming in, where there is depth sensitivity, where there is a artificial cover is there. So, suppose if you are talking if there is a lot of loose material, soil is there, a lot of then those kind of measurements becomes very has a lot of error in it, okay. So, finding matching it back to calculating it back and finding out the errors, that can then become a little bit problematic with the remotely sensed data sets. So, given these advantages and these disadvantages, there is another kind of data that is of course a lot more preferred in aquatic geochemistry, and that is the ground observed data. So, even though we use a lot of remotely sensed data, ground data always has to be collected and validated with the remotely sensed data. If the remotely sensed data lacks validation, then most of the times it cannot be used for any kind of scientific research purpose, okay. What is ground observed data? That means you are collecting the sample from the particular area, that is field based sample. So, from that river water or the lake water, somewhere you are traveling, you are going physically collecting the water, you are analyzing the sample and you are interpreting the data. So, this is my ground observed data that starts from sample collection to my sample analysis, and therefore, what is my interpretation. Now, how ground observed data when we are in aquatic geochemistry is extremely important, and it is almost as important as I would say as your data interpretation is, because if during the process of sample collection you make a mistake, okay, then it then it will translate into your data. You will be making an erroneous measurement, you will be then not be able to make the rightful interpretation. So, it forms the first step in trying to address the research problem that you have decided, and a lot of our focus and a lot of our attention has to be given in this sample collection, sample analysis, and how the data is going to be interpreted. A lot of energy actually goes into this. Now, when I say that you will if you want to collect a groundwater sample, will you be able, if I give you a project, then will you just simply go and collect a water sample from anywhere? No, right, you always have to have some background work done, some kind of selectivity has to be done on where to do, how to do, in what frequency it has to be done, what all other data is required. So, all kind of things need to be kept in mind when we are going for a sampling field work. And if I break down this, okay, if I break down this, then first we have to first we have to select the place where we want to sample, right. We have to find the proper place where we need to take the sample, which will give us the rightful the right kind of data that would help us solve that kind of problem. Type of sampling, how we are going to do sampling. I will talk about the type of sampling in the further slides. Collection methodology means how we are collecting the sample. There are different methods of sample collection. So, that sample preservation, every type of analysis they can have particular type of preservation required. Other required data they can be in situ data, ex situ data, and these are our simply the data sets that we need to add in more information, and without that information it becomes incomplete. So, to have the entire picture we need to have these kind of other data as well. So, therefore, this in situ, ex situ data and corresponding data sets. And of course, an extremely important aspect is ethical and safety aspects. While doing a particular, while trying to address a particular problem, you yourself cannot prepare more problems. Okay. So, there are ethical issues, there are safety issues, those things have to be given importance. A few types of chemical analysis I would be definitely be talking about, and the most important ones I will be definitely discussing about. And of course, then data interpretation is extremely varied, it is as varied as the types of problems that is being. So, every problem has to have a proper interpretation. It is basically a tailor fit, it varies with each problem, not there are some basic things that is taught. After that, to find out the solution you have to you have to tailor fit your data, and you have to tailor fit your models, and whatever kind of analysis and interpretation techniques that you are using, you have to develop that. So, moving again, as I said in the very beginning of the lecture itself, that this particular course is about natural surface waters. So, that is oceans, rivers, lakes, and the type of sampling depends on our research. That is, what are we collecting? Is my research related to very long term observations and all? Will it be useful if I conduct my research in oceans? Am I interested in doing very long term studies? Then we have to look into research in oceans. So, the chemical oceanography we should be looking there. Or if I am looking into terrestrial ecosystems, I may be mostly looking into lakes, groundwater and rivers, right. So, the type of science that I want to do, what kind of water are we collecting, what kind of sampling we are doing, that depends on this kind of this the first basis is what kind of research objectives we are having. And again, so these are again some examples of natural subsurface waters. So, groundwater, there is pore water. Sometimes we need to understand how the pore water looks. So, in between the grains, as you can see, there is always, not always, but during after a rainfall event, there will be pore water there, and even the top soils almost always will have a little bit of water. So, that is the very tiny amount of water will always be there in the topsoil. So, that is called the pore water, right, and that is coming due to the capillary effects and all that. So, the groundwater will be pulled up. So, it will be due to the capillary effect that pore water will be there in the soils. And other forms of water. So, there we can work on, anybody can work on rain, some people are working on glaciers, snow, ice, people are also working on fluid inclusions in rocks. Okay. So, in rocks, when it is forming, there is a maybe there is a tiny little bubble created and a fluid droplet gets stored in that tiny bubble. So, that tiny bubble the people have to extract that little bubble of water. It can actually be a very small volume of water, and you sometimes will be amazed to see how much small volumes of water we can extract, and we do studies on them. And these are again very rare kind of studies that has to be done to understand sometimes as old problems like related to rock cycle, related to mantle geochemistry, related to earth's historical processes, and even samples coming from space in meteorites and from other planets, samples that is being collected from other planets. So, those kind of studies are done on fluid inclusions. Hydrothermal veins. What are hydrothermal veins? Volcanoes, I think you have heard about this term called volcanoes. So, volcanoes are when there is a break there in the tectonic plate and magma forces itself out through that break. So, either it can be a joint between two tectonic plates, or it can be a weakened lithosphere out there. So, some opening that releases the deep magma from the internal inside of the earth, and that gets released onto the surface. Now, that is the observation that we are making on the surface. What is happening inside the oceans? Now, oceans also, as of course, 3/4 of the Earth's planet is covered with oceans, right. So, there are many joints, tectonic plate joints in the oceanic under the ocean. Now, along these joints there will be these weak weak points, weak joints. These will be releasing a lot of heat, a lot of material from inside of the earth into the water. So, these are called hydrothermal vents. Okay. Hydrothermal because they are hot water, these are actually hot water vents. Okay. Then planetary water, comets, etc. We can go on and finding the types of water that is found in from space. So, basically this is the kind of natural water that we are going to talk about in this particular course. Now, moving on to contaminated waters, of course. So, many times we also have to work upon contaminated systems, and that is one of the most critical thing to work upon, because it is actually a problem and now it is more than scientific, more than to satisfy our scientific need, we have to understand the need for the safety of our ecosystem, and we have to address the problem. So, anywhere where sewage from the industries or from the domestic sewage or from urban sewage, that is going into a particular, that is getting discharged into the lake or the river, or sometimes it can seep into the groundwater, how that contaminated water has to be dealt with, what solutions can we bring. Mining effluents, okay, mining effluents again are of very different compositions because of the minerals that it is dissolving, of the kind of exposure that has been, it is being exposed to, that controls the mining effluents, and how that effluent will be released into the natural surface water and what impact can it do, that has to be constrained. Now, what constitutes water? Now, whenever we are taking a sample, it is not that it is only water, right. So, any sample that we are collecting, it can be broken down, it can be broken down into something which is absolutely dissolved. So, the constituents are absolutely dissolved in the water. So, that is called the dissolved load. And when we are filtering, if we find things like this on the filter paper, then this is called the suspended sediment load, right. So, this is the suspended sediment, and of course, there is the bottom or the bed sediment. Now, this is an image which shows at so now how much what is getting dissolved, how much is getting retained in the filter paper, okay, all this is determined by what is our filter paper pore size, okay. So, there is actually particularly 3 phases. So, we will start from here, that is the particulate phase, right. So, particulate phase are the larger than whatever is larger than this 1 micrometer. So, sometimes in the olden days we used to have this as our filter paper pore size. Now, as we are moving towards having more technological innovations and all that, now our particulate pore paper size delineation has reduced to 0.2 micrometer. Okay. So, if you want to separate out 2 phases, that is the particulate phase from the dissolved and the colloidal phase, you are going to use a filter paper with a pore size of 0.2 micrometer. Okay. Now, there is another phase called the colloidal phase. Now, what are colloids? They are mainly complex molecules. Okay. So, these are larger molecules, these are complex molecules. So, these some metals, they prefer to be in the colloidal phase. For example, iron, it is an extremely important element in terms of understanding and controlling water and its quality, and this likes to remain in the colloidal phase. Again, how do we define the dissolved phase? So, whatever is passing beyond this 1 nanometer filter pore paper, that is called the absolutely dissolved phase. So, it is a very tiny pore size, just imagine 1 nanometer in scale, and whatever is passing through that filter paper, we are calling that is my dissolved load. So, here you can see this is a syringe filter where the filter paper is rolled inside this tiny glass, it is not a glass tube, it is a kind of very clean plastic, and this this is the sample that has been collected. It is pressured through this rolled filter paper, and then the clean and the the filtered water is collected in the bottle. So, this is what is division between the inorganic compounds and the organic compounds, and you can see the size differences. So, clay, carbonates and phosphates, they are of bigger size fraction as compared to the colloidal hydroxides of iron and manganese, and some of the most of the elements in their hydrated cations and anionic forms, they are mostly in the absolutely dissolved phase. In terms of organics, amino acids, peptides, proteins and fulvic acids, they are absolutely dissolved. As colloidal, in the colloidal scale you can find viruses, some polysaccharides, that is a form of carbohydrate, and humic acids, complex humic acids, larger molecules, they remain in the colloidal phase. And the larger, in the particulate phase you will mostly find algae, you will mostly find bacteria and cell debris. So, we want to separate out, because when we are going to analyze, we want to know the true nature of water. That means, the water that is going to be uptaken by plants, that is going to be uptaken by animals. So, we need to separate them out to understand what is there in the truly dissolved form, and whatever is there in the suspended sediments, whatever is there. See, as you can see that the suspended sediment, right, will definitely have some be made up of some clay minerals and all. So, they will have a very different composition, right. As you can see here, the bottom and the bed sediments, they are very coarse in nature. So, these these tiny, these rocks, they will have different kind of minerals, and if you try to analyze them, then they will have a significant concentration of many different metals. So, we are not going to use those values, we are going to use the dissolved load values, because this is what is going to be bioavailable to the plants and to the animals. Now, what is the importance of filtration, right? Why do we need to filter? This kind of studies has been established in the past, and from many decades it is in common use and common knowledge, and this kind of experiments were done. As you can see, this paper was published in 1974. So, what they did is, in this particular axis, as you can see, is the concentration of particular metal given in microgram per liter. Okay, how do you denote it? We denote it in the form of microgram per liter, and on the x axis we have different filter paper pore sizes, and we want to understand what will happen. First of all, if I do not filter, if I filter it with different filter paper pore sizes, what therefore we need to understand the importance and the attention the filter paper pore size needs when we are doing our research. And two metals has been selected to show you the difference. One is aluminum and the other is iron. Okay. There are two curves, okay, and it is said here, it says that the first analysis was done after 19 days of sample collection, right. So, a water sample was collected, it was analyzed, okay, it was filtered and it was analyzed after 19 days. Okay. So, it was filtered using these three pore sizes. So, the same sample was filtered using three different filter paper with different pore sizes. One pore size was 0.45 micrometer, the other was 0.22, the other was 0.10 micrometer. Okay. After 19 days, what do we find? We find that the water that was filtered using 0.45 micrometer showed around 45 microgram per liter of aluminum. The one that was filtered using 0.22 micrometer had a concentration of around 23 or 4 microgram per liter. Okay. And the one that was filtered using 0.10, that one had the lowest concentration after 19 days, that is around 11 microgram per liter. Now, what happens if that same water was allowed to stay and analyzed after 94 days. Okay. So, after 94 days, the water was found to be around 60 to 3 microgram per liter. Same sample that was analyzed after 94 days, it was found to vary little higher for the sample that was filtered with 0.22 micrometers, so around 28 or 27 microgram per liter, and this one after 0.10 sample, this sample was again a little bit higher. What does this show? Right, what does this trying to imply? And the same thing for iron as well. So, one set of sample was analyzed after 19 days and one set of sample was analyzed after 94 days. Okay. And this is the difference in their concentration within that time period. So, it means that the sample that was filtered using 0.45 micrometer allowed to pass some of the finer, very finer particles. They continue to interact. Okay. They continue to interact over the 94 days period, and after 94 days when you are measuring, you are finding the aluminum concentration to be very high. Right. Same thing for iron. Initially, after when the first analysis was done, that is after 19 days, the value was 40, sorry not 40, 36 or 7, but after 94 days it was considerably high. It was quite close to double of the initial value, that is around 64 or 65 microgram per liter. And as we lower the filter paper, okay, as we lower the filter pore size, the finer particles are not allowed to pass through. Okay. That means the finer particles, they get stuck into the filter paper and they no longer interact and change the dissolved concentration. Therefore, the one with 0.22 sample and the sample which has passed through the 0.10 micrometer filter paper, both of them showed that it was similar. Okay, they are quite maybe 1 or 2 microgram per liter difference is there. Okay, but it is minimal. It can always be said that it fall within the error and such things. So, you need not worry. Except for iron, in case of this, that means it shows that for iron the colloidal phase can also be a significant contributor, and if you want to do much in depth analysis for iron, you may have to actually use this filter paper pore size, because this completely retains back, separates absolutely the finer and the colloidal phases from the true dissolved phase. Right. So, this actually plot shows you the importance of selection of the filter paper pore size. If you are using a much higher filter paper pore size, something like 0.47 or 0.45 micrometer, then you expect that some finer materials will be passing through that filter paper pore size, and that if you allow it to remain for a much longer time period, then it might show changes in the concentration of the particular elements that you are interested. So, with this particular information, I will end this lecture of today, and we will continue from here in our next lecture related to the water sampling techniques. Thank you so much for joining.